So I'm going to talk a little bit about what CRISPR is, give you a little bit of scientific background on what it is I want to talk about today, and then go into a little bit more depth on what are some of the questions that this raises from a position of society and what kind of society we want to live in. That one. So as was already mentioned, I work for the Innovative Genomics Institute. We were founded by Jennifer Doudna, who is one of the co-inventors of CRISPR technology. And she founded the institute to develop and deploy genome engineering to cure disease, ensure food security, and sustain the environment for current and future generations. So essentially, we are a scientific research institution. But something that's been really core to the foundation of this institute was the realization that this is not just, a simple lab technology. There's actual real impact that this could have on the world, both for the good. So, we're trying to use it for good. We're trying to deploy it to cure disease and ensure food security and sustain the environment. But there may also be negative consequences of this. And to really understand how it should fit into society and how we can think about it and best use it, we need to be simultaneously thinking about the societal impacts as we're doing the research. So we've also founded, along with this, a research program in society where we fund ethicists, lawyers, humanities, doing research on what are the ethical, the legal, the social implications of this work. So that's a little bit about the Institute, but I want to make sure that we're all kind of on the same page before we talk about what this is. So I know that people are here from all different backgrounds, so let's take a step back. I don't know how long it's been since you've had a biology course, but we'll do a quick overview. So some terminology. So you can think of a genome kind of like a book, and this is an instruction manual of all the information that it takes to make you. And then chromosomes are a way to organize that information, just like chapters are a way to organize information in a book. And sentences can be thought of as, genes can be thought of as sentences. So within those chapters, there's a start and an end to each gene. Each gene is a self-contained bit of information that has one instruction for one part of making your cells. And then the language with which these sentences are written in is a chemical language called DNA. And I'm showing here the 4 base pairs, so chemical bases that are used to make up the sequence, the language of this DNA. So the order in which these come dictates what the meaning of the sentence is. So that's a little bit getting us all on the same page of what are genes, what am I talking about when I say genome editing. So what is CRISPR? CRISPR is actually a naturally occurring system. It was originally discovered about 30 years ago, actually, which may come as a surprise because most of you have heard of it fairly recently. The CRISPR itself, the system comes from a bacterial defense system. So it turns out that viruses infect bacteria. Just like we get infected by viruses, bacteria get sick. And what I'm showing here are an electron microscope, scanning microscope image of a bacteria in green. And then all of these little viruses are pseudocolored in pink. And you can see that they attack these bacteria. They sit on the surface, and what they do is they inject their DNA into the bacteria. Then they hijack the bacteria's system to make more viruses inside and eventually explode out. So here you're seeing this horrific explosion. So over the millennia, viruses and bacteria have been in conflict. They've had this battle. And they've both been evolving ways to viruses better attack bacteria and bacteria better combat the infections. And so CRISPR is actually a result of that evolutionary battle between the two. So here I'm showing you a little image of bacteria. Just like all living things, bacteria have DNA. So here's their chromosome. And CRISPR refers specifically to this region of DNA within the bacteria, which is a molecular memory of these infections. And the way it works is that a virus comes. When it infects the bacteria, it injects its DNA in. Now, most of the time, the bacteria will die. But they have multiple defense systems. And every once in a while, you'll have some that survive. And those that survive will find the DNA from the virus. They will make a copy of it using this memorizing complex and insert it into their own genome. And they do this in a location called a CRISPR array. So each one of these different colored bits of DNA represents a copy of a virus previously infected, most likely from a previous generation. So now that this bacteria has survived the attack of the viruses, it will pass on that genetic memory of a viral infection to the next generation of bacteria. And in this way, you have this molecular memory of invading viruses, so that the next time the descendants of this survivor are exposed to the same virus, they have a way to recognize it and kill it immediately before it has a chance to fully infect it. And the way they do this is they copy the bacterial chromosome. They take a little teeny copy of this section, and we call that a guide RNA. So RNA is essentially a transportable version of DNA. They load it into this thing called Cas9, and together they make this cleavage complex. And this complex is constantly floating around the bacterial cell looking for a match. So the guide RNA is kind of like a molecular mugshot of the virus. It's constantly searching for an exact match. And when it finds it, it will bind and it will cut. So let's see. So the thing that we've all been hearing about more recently is not so much this, because we knew a lot about this over the last 30 years. It took quite a while to figure out exactly what it was that this weird sequence was doing, but we started to realize that there must be some kind of molecular memory happening. The thing that's really transforming the way we do research, and the thing that I'm talking about today, is the way that we've actually repurposed this system. So these two people had this moment of realization where they were just studying, basic science, trying to understand this very cool system, because it turns out bacteria have immune systems. How weird. Normally you think that it's only you that has an immune system. And what they found is that the way that this match happens is very straightforward and simple. It's just literally the exact sequence of this binds to the exact sequence of that. And so what they thought was, well, what if instead of having that sequence match a virus, what if it matched something that we want it to match? What if we took this whole thing out of the bacteria, we changed this sequence, and instead of a mugshot of an invading virus, maybe we gave it a copy of a malfunctioning gene that's causing a disease? Would it be able to then go and find that malfunctioning gene and repair it? And it turns out that, in fact, almost shockingly, it does. And it works incredibly well. So why would you want to do that? So if we go back to the analogy about a book in sentences, we have here a gene that I'm showing, and in its normal state, it has an A at this location. And maybe this means twinkle, twinkle, little star. But every once in a while, you have a mutation. Sometimes these are inherited. Sometimes these are caused just by exposure to UV rays and various mutagens in your environment. And when that happens, now you have twinkle, twinkle, big star. So sometimes a very small change can have a very significant impact. And this is actually the exact impact that happens in sickle cell disease. So sickle cell disease is a disorder of the blood. It's inherited, and it's caused by a mutation at a single gene at a single site. Why is that doing that? Where it goes from an A to a T. And that is enough to cause a malformation of these red blood cells into the sickled shape at the bottom. This is eventually lethal. It's a very slow, very painful experience where you are essentially depriving your organs of oxygen over your lifetime. And people that have sickle cell disease have a life expectancy in the US of their early 40s. So if we can have a way to target it, we can cut the location. So editing always begins with a cut. It goes to the site, it binds, and it cuts. And then the edit is actually something that happens inside your cells naturally. So I mentioned that there's mutagens in your environment. Your DNA is constantly getting bombarded and destroyed. So we've evolved mechanisms to deal with this. Your cell has two pathways that it uses. One is where it just randomly tries to stitch the two pieces back together. And most of the time, just blunt end stitching together works fine. But when you have a CRISPR constantly binding and cutting, you start to push for misrepairs. And only when it's incorrectly repaired does it stop binding and cutting. So some small percent of the time you have a mismatch, and now you end up with a disruption in the sequence. And you can think of this as knocking it out, as essentially deleting it. When it's not written correctly, the cell usually just doesn't use it anymore. It stops working. The other method is to provide the cell with a donor DNA. So we have two copies of DNA in our bodies, one from our mom, one from our dad. And it's very useful if one of them gets damaged to have that second copy, because you can find it, you can use the information in that sequence to properly repair the broken copy. So scientists are taking advantage of this by, instead of having the cell repair it using the sister, that's what it's called, the other chromosome, we can give it something that looks a lot like the location of the repair on either side, but has the thing in it that we want to insert. So this might be the A instead of the T at the sickle cell location. So in this way, you can delete, so knock something out, or you can add something or change it very discreetly. So this is, in fact, something that we're doing currently, trying to address sickle cell disease. And the idea would be that you would take these blood cells out, you would isolate the progenitor cells. So these are blood stem cells that repopulate blood. You would fix that mutation and then reinfuse it back to the patient. And over time, those blood cells, your blood is not the same in your whole life, it's constantly being turned over. So over time, the corrected mutation in those stem cells will repopulate the blood of the patient, and they will effectively be cured. So these are really exciting things, and it turns out, everything that lives has DNA, and everything that we've tried CRISPR on so far, it gets edited very easily. I should say easily with an asterisk. It's not as straightforward in everything as you might think, but we've been successful so far. So we went from a time where we had just a few model organisms to study disease to basically anything you can think of. These are all things that have been edited at this point using CRISPR. And this is in the span of a few years. CRISPR was first discovered as a molecule that you could use to edit DNA in 2012. So this is 7 years. This is where we are. We have preclinical approval for certain gene editing for various diseases going into the clinic. So this is quick. So this is all very exciting. But why am I here telling you about societal implications? So there are a lot of really exciting things that we can do, but this is very powerful technology. Because we can use it on anything that has DNA, there are many options of things that we could do that we can manipulate them. And broadly speaking, there are a few things that we have to think about across the board, no matter what we use it on. One would be equity and access. So concepts of social justice demand that burdens and benefits are distributed fairly. So the sickle cell example that I showed you, is everybody going to be able to be cured by sickle cell? How are we going to pay for this? It turns out that the precursor to gene editing, which is gene therapy, we have a few of these coming on the market now, and their price tags are in the order of close to 1 million, if not above. So the most recent one is treating a disorder, lethal congenital disorder, and its price tag is $2.1 million. It cures it, it's one time, and they survive, but how do you pay for that? So this is very promising, and it's estimated that potentially the sickle cell treatments may be in the range of $1 million a pop. So people are thinking about, on one hand, you're saving money by treating people who would otherwise be hospitalized over the course of their life. So there is some equity in the charge there, but this is happening up front. Are you going to be able to have insurance companies cover this? And if not, who are going to be the people that actually use this? What are the treatments that you're creating? Are you creating treatments for the most horrifying, debilitating diseases for the most people, or for the people that can pay for them? Are you even treating diseases at all? Perhaps you're looking at something that gives somebody an advantage, or something that's more exciting, but it's going to cost a lot of money. So who's going to be able to use that? I'm going to skip ahead real quick to touch on one last thing in the few minutes that I have. One of the biggest questions that we have facing us with gene editing is not just treating existing humans, but treating or manipulating people before they exist. So this is something called germline editing. And the distinction here is that somatic cells are cells that have developed, they're part of an existing person, and they're what we call generally terminally differentiated, which means you can't take this cell and create a whole new person from it. Germ cells you can, and in fact these are things like eggs and sperm and embryos, and even the cells that create the eggs and sperms and embryos. So why not, rather than treating a sickle cell patient, we know it's inherited, let's treat the embryo, get rid of the sickle cell disease before the person's born. That seems like maybe a logical thought. But there are a lot of reasons that this might be concerning. There's a lot of risks that we have to consider. So I'm categorizing these here in roughly 3 categories. We have technical risks, philosophical risks, and societal risks. So what do I mean by those? So CRISPR is extremely powerful. It's very precise. It's quite accurate, but it's not perfect. It's biology. Biology is not perfect. So some of the time, rather than targeting the correct location, we get what we call off targets, where it will cut somewhere that it's not supposed to. Now, if you do that in a progenitor blood cell, the mistake is only going to affect the cells in the blood. But if you do that in a germ cell, that mistake is going to affect the entire human and all of its offspring. It also is important to know that while we understand a lot about the genome, we don't understand everything. So to be able to predict what those off targets are going to do to the development of an embryo is actually very challenging. Real quick. So there's other issues, technical issues here. One is what we call mosaicism. So the thing that people are talking about doing is generally editing at the stage of in vitro fertilization. And then you have a clock ticking. So if you add the CRISPR in at the same time as the sperm and the egg, you have between the time that it hits the egg to make that edit faster than the zygote starts to divide. So if the zygote starts to divide before the CRISPR has a chance to actually get in and make that change, then some of the cells are going to have the edit and some won't. Okay, so this creates what we call a mosaic individual. Again, this is something that we can study in model organisms, but it's something that we really don't understand well for humans and what kind of consequences might be there. And this is something that is not reproducible from person to person. This is random. So you're not going to be able to know which organs are going to have which mutation before it happens. So those are the technical risks, and in theory, those are surmountable. I should also mention that any kinds of medical treatments, we do clinical trials. We try to do it carefully. How are you going to do a clinical trial for creating a person? Who are you going to enroll for this? You're going to create a certain number of individuals, and then you're going to track them for 80 years. You're going to have an 80-year clinical trial. It's actually technically very challenging to think about how to do this. And I'm running out of time. So, but there's also other risks that we need to think about. So there's philosophical risks. A lot of people immediately jump to religious objections, and we should take these seriously. But there are also things that are not religious-based. So a lot of people, whether they're religious or not, have a sense of what it means for an embryo to be changed, that that's creating a person, and that there's something really heavy about that. And it's not that you are treating an individual that has a disease, but you are creating somebody in order to treat them. You're creating somebody to treat. And then there's also questions of treatment versus enhancement. And I'm showing here, debilitating disease versus something maybe increasing your muscle strength so you can be a really fast runner, right? That would be enhancement. But there's a lot of things that lie at the middle there. A lot of people think enhancement, no. Treatment of debilitating disease, sure. But where do you draw that line? So for example, there's a gene PCSK9 that confers resistance to heart disease. Is that treating or enhancing? If you give that mutation that's found at a very rare percent of the population and it makes them have a stronger heart, are you enhancing somebody or are you treating somebody with that? There's a lot of gray area in here. And then the last thing that I think is actually maybe the most important, in my opinion, is the social risks. So what happens if we do start treating embryos and germline cells? Who are, again, going back to the cost, who are the people that are going to be able to do this? For the foreseeable future, it's going to be the wealthy. This is not going to be something covered by insurance. This is going to be an added cost onto an already very expensive in vitro fertilization procedure. There's also questions about identity. So how do we identify with our genes? At what level are we us, and at what level are we our genes? We should think as well about class structures. So again, because of the access issue, you're going to have a class of entire people that have access to, whether it's true or whether it's in theory, they have improved genetic children. So now you have creating an entire class of people where the perception is that they at least are genetically superior to others. So that you're starting to get into the realm of eugenics, which concerns a lot of people. So with that, I'm going to show this last slide. So this has led to a lot of people trying to grapple with these issues. I'm showing here a list. This is the results of National Academy of Sciences consensus study, where they tried to think, is there a pathway here that would allow an ethical use of germline editing? And here's kind of the list that they came up with, things that would have to be satisfied to allow germline editing. And it's not clear yet whether any of these have been satisfied. Regardless, though, the first babies have been born that have been CRISPR'd. So this makes us start to think, how do we move forward? How do we ensure that there's safe use here? Is the cat already out of the bag? Can we not put it back in the box, so to speak? And how should we proceed? So these are all questions that, as a species, we should be deciding together. We should be thinking about together. So that's it for me. Thank you.